Okay, so get microphone should work. Yep, and I'll just maybe lower the lights a little bit more.
Okay, good morning, welcome everyone. I'm Francisco Sinasviso from CSRO, and I have the pleasure to introduce today the speaker, Camille Roux. And Camille is an evolutionary biologist. He did his PhD in Lille in France with Xavier Beckerman in evolution of plant mating systems. And after that, well, he did postdoc in Switzerland, and France, and UK. And now he's a CNRS researcher in Lille, also in France. He likes Lille. It's a nice city. And he studied evolution of species. I mean, obviously, speciation, molecular evolution, and evolution of plant mating systems. And, well, he has today he's going to talk about speciation in dynamics in plants and animals. And I'll give the floor to Camille. Thank you.
Hello, thank you. Do you hear me? First of all, I would like to thank you because I've never been so well received actually. And I'm not saying that to be polite, so thank you. So I will talk to you today about a work made by François Monet, who is my PhD student. And we wanted to do a kind of comparison between the dynamic speciation in plants versus animals. Does that work? Okay, I can click. Okay. I will not do a big introduction about speciation because it's a quite complicated process, but some generalities. One of the biggest generality, of course, is that we expect a kind of a general relationship between the age of the common ancestor of two lineages. So here you can see the tree of life. It's a very broad scale. With here in brown some archaea, in green the plants, in dark green reptiles, here the mammals, etc. And there is here a kind of general relationship between the age of the common ancestor and the amount of reproductive isolation. Regardless of the nature of the reproductive isolation, we expect that if the ancestor is old, we have many species barrier. Okay. I took this very simple example. It's one I studied when I was close to Montpellier. There's some two sea urchins named Abatus cordatus. Okay. They share a very recent common ancestor. You can find some populations in Spain, in Greece, and because of this low level of divergence, we have a low level of reproductive isolation, and because they are physically connected by migration, so they can produce some hybrids, and these hybrids are fertile and have basically introgression. And of course, in an extreme, the common ancestor between the coral and sea urchin is much older, and here we have a fully we have fully isolated species, and even if they share the same geographic area, well, we do not find any hybrids, of course. So this is a process of speciation. It's an accumulation over this history. It's an accumulation of a specific kind of mutation that we name species barrier. And this species barrier has a specificity to reduce the thickness of the hybrids. Okay. So we have different outcomes due to a migration of a species barrier. Here you can see two gene pools, gene pool A, gene pool B. Here we have a very simplistic model of a species barrier. Species barrier is just a black dot. And during a migration event, the migration event is a physical transfer of one chromosome carrying this species barrier from population B to population A. And from this situation, you can expect different results. If the barrier is very strong, or if there is a very low recombination rate around this barrier, well, we have the complete rejection of genome B from population A. So we have no introgression because the barrier is very strong, or because the recombination rate around the barrier is very low. But if the barrier is weak, or if there is an elevated recombination rate around the barrier. So here we will have the rejection of the barrier. And so we can have these neutral regions coming from B that will escape through recombination. They will escape to their linkage to the species barrier, and so they will be established into the population A, and this is what we call introgression. And of course, if all of the genomic positions are statistically linked to a species barrier, well, introgression is impossible. So introgression is this continuous process. It's a continuous process of accumulation of species barrier. And this continuous process leads to two discrete entities named A and B. This is A, this is B. So it's a continuous process, but we can try to roughly subdivide this process in 3 steps. In the first steps, at the origin of the speciation, actually. So just after the subdivision of one ancestral population in 2 daughter populations, A and B, in this case, the divergence is recent. And because it's recent, we do not expect any species barrier. And so all of the genomic positions are expected to share the same probability of being exchanged from A to B or from B to A. Basically, we have no barrier. So we have a kind of genomic homogeneity in introgression rates. And this is why what I call populations from the same species, because they have no barrier. But after a given amount of time, when the first barrier starts to be well established in gene pool A and gene pool B, here we have the effect of this barrier. So here, the gene flow is no longer homogeneous. We have a genomic heterogeneity in species in introgression rates. So in this case, A and B, well, they're still able to produce some hybrids. So we cannot call them species because they produce hybrids. But they're still different, actually. So they're not populations. This is what we call semi-isolated species. So basically, semi-isolated species is one intermediate case. It's an intermediate status. And at the end, speciation is over when we have many species barriers throughout the chromosomes. So during this project with François Monet, we were interested about the dynamic of speciation. We can summarize by 2 simple questions. When occurs the transition from populations to species? And when occurs the transition from population to semi-isolated species? So when occurs this transition and this transition? And finally, is there a threshold of divergence above which gene flow is impossible? So speciation is a quite complex model, so a quite complex process. There is no integrative theory, no integrative model in order to predict the variability of the hybrids. What we can simply do is it's an empirical exploration of a continuum of divergence. So what we do is simply exploring a continuum of divergence made by multiple independent pairs of A and B, and trying to see if we have migration here, migration here, migration here, migration here. So it's a very simple project. And more specifically, we take some gene pools in nature, some pairs of plants, some pairs of animals, whatever. We measure the divergence in order to see where we are along this gradient of divergence. And we also do some model comparison in order to see if we have recent introgression or complete isolation. That, of course, we do not have a direct observation of the process of speciation. We do not have a direct observation of introgression, but we have genomes. So here it's a genome, here, and I represent here's divergence. And over the genome, you can find some genomic regions for which A and B are very similar, and regions for which A and B are very divergent. So we have this kind of heterogeneous pattern of divergence along the chromosome. So the goal is to try to detect some signatures of introgression along this chromosome. There are multiple verbal models to explain this pattern. So it's very easy to explain everything by any models. And this is what many people used to do between 2010 and now, just to take an observed genome, to take a favorite model, and to tell the story. So verbally, we can say that, okay, I want to explain everything by this model. We have a model of isolation with migration, the gene flow. Gene flow will maintain a very low level of differentiation throughout the chromosome. So here under this model, the only way to find some peak of differentiation is to have some strong local adaptive processes. And if I want to explain everything by this model, this peak will be necessarily some selected loci. If I hate the models of ecological speciation, I will explain everything by that. So here, at the beginning of speciation, we have a very low level of differentiation, of course, and over time, the differentiation will increase progressively throughout the genome. But some genomic regions, we have an accelerated differentiation. It's the case of the background selection, we increase the differentiation, and it's also the case of selected loci. This model is not compatible with local adaptive processes. And finally, we can also explain everything by this model of secondary contact. So here we have an ancestral population subdivided in two isolated populations. During this phase of isolation, you increase the differentiation everywhere. So you fix some neutral alleles, but you also fix some species barrier. Oops, some species barrier, yes. And during a secondary contact with gene flow, you reduce the differentiation in the genomic regions which are not linked to a species barrier. And tada, we verbally explain the same model. So this is why we need a statistical framework in order to quantitatively compare alternative models of speciation. So we take this genome. In practice, we take this genome. We can summarize this genome by using n statistics. So here, just for the illustration, we summarize it by the mean FST and the genomic variation for the FST. Okay. And I want to statistically evaluate my model IM for isolation migration. So what I do, I'm just doing a large number of random simulations under this model. And over each of my simulations, I compute the same statistics than statistics used to describe my observed data set. Like that, after many simulations, I can empirically approximate the joint distributions of my statistics. So I do that under this model. Under its competitive model, DSI, the same work. A large number of simulations under this model by taking random combination of parameters. We compute the same vector of statistics, and then we empirically approximate this joint distribution of statistics. And the final step is a statistical comparison between the observation and the simulations. So in this case, it's obvious that ABC will provide a strong statistical support for the model with migration. In this case, a strong statistical support for isolation. And in this case, well, we will have an ambiguity. So it's one advantage of ABC that it's very rare to observe false positive, but we have this kind of ambiguity, but we can detect them because they have intermediate values of probability. So it's cool. I prefer to have a very low rate of false positive. So okay, let's go back to my questions. I want to understand my dynamic of speciation. When do we observe the transition from populations to semi-isolated species? So when do we observe the first barriers? And when do we observe the complete interaction of gene flow? We have a continuum of divergence. We have the models that we want to estimate. Some of the models have ongoing migration. Some of the models have current isolation. So the goal is for each pair of population of species is to compute the probability of that versus that. And if we have migration, either migration can be homogeneous or heterogeneous. So either we have populations or semi-isolated species. We did that. All of this statistical framework is implemented in a tool named DILS, something that we published 2 years ago. It's easy to handle. You upload your data, and you have the probability of models of migration, of isolation, and you also have the probability that you have populations or semi-isolated species. So we ran DILS, just some detail. So here it's just some old results that we published 7 years ago. We did that in animals. So, and after that, I will show you the results in plants to show the comparison between plants and animals. So in animals, we had 61 pairs of animals, so 10 pairs from the NCBI, and 51 pairs that we sequenced in a natural population. And we did basically RNA sequencing. So we done some pairs of invertebrates, some pairs of vertebrates. All of them are non-model species, which is important. And importantly, we choose them with no a priori about the process of speciation. We do not choose them because, well, maybe they're sexy for introgression or not. They are anonymous pairs for us. We randomly took them, and that's it. This is the explored range of divergence. So the divergence here is measured as the percentage of the genome which is different between species A and species B. Basically, human chimpanzee, it's 1%. Okay, here you have human chimpanzee. So we explore the range of divergence between very low and 30 times human chimpanzee, and here is just the FST. And here it was the result of this study. I represent this dynamic of speciation as follows. On the x-axis, you have the divergence, 1% human chimpanzee, just to have a kind of reference. And on the y-axis, you have the posterior probability of models with ongoing migration. In blue, you have the pairs of population or species here. 1 point is a pair, 61 pair. In blue here, you have the pair of gene pools for which the ABC approach provided a strong statistical support for migration, and this migration is homogeneous, no barrier. In purple, pairs of A and B, so pairs of gene pools with a strong statistical support for migration, for ongoing migration, but this migration is heterogeneous. Some genomic regions are associated to species barrier. And in red here, you have the pairs of species for which we do not find any support for migration. So we can call them fully isolated species. So it's a very simple way to represent this dynamic of speciation. And what we can see is that, okay, the first barrier, this one, appears quite rapidly at 0.08%, which is much below than most of the levels of diversity in species. It's very low. So we can detect the first barrier very rapidly. Something, because when we explain this three states view of population and species and semi-isolated species, it looks obvious. But most of the time, the statue of speciation is binary, population or species. And there is this kind of ambiguity. Okay. If we are intermediate, how can we call them? So the solution for me is just to call them semi-isolated species. When we do a meta-analysis, it's funny because we have a lot of semi-isolated species. So they are fully neglected, but they are prevalent, actually. So don't neglect the semi-isolated species. And it's something that we name the gray zone of speciation. It's this segment of the continuum of divergence in which you can find two different situations. For the same level of divergence, you can have situations in which you have gene flow and situations in which you have isolation. So sometimes 2% of divergence is enough to completely interrupt gene flow. But like here in muscles and in coral, well, they don't care about this 2% of divergence. They are still able to produce some hybrids, and these hybrids are viable. But here it's 2 times more divergence than human and chimpanzee. Okay. So this is this gray zone between 0.5 and 2%. And after 2% of divergence, in this analysis, we did not find any case of introgression. So when we did this analysis, for me it was like, it's funny because 2% of divergence, it's what we can read in the old papers by taxonomists. They put these 2% of criteria to delineate species. Actually, it works well. And I show that to a lab in microbiology. And these guys, they tried to do some experimental introgression of a plasmid in bacteria by increasing the level of divergence of the plasmid. At 0 level of divergence, you can introduce your plasmid, etc. And after 2% of divergence, they are not able to introduce their plasmid into the bacterial genome. So for me, it was, it's funny, we have a kind of universal threshold. Actually, no. So I can summarize this figure like that. Okay. Here we have the populations. Here we have semi-isolated species. And here we have fully isolated species. And now I want to study the plants. Is it different or not? So we can have different expectations. Either plants exchange genes for higher levels of divergence than animals. In this case, actually plants exchange more easily genes. We would expect a kind of decay here. We will expect that populations until higher levels of divergence for plants. We will expect more of semi-isolated species. No. Maybe it's the opposite. Maybe reproductive isolation evolves faster in plants than animals. So in this case, in plants, we will observe a shift to the left. Okay. So maybe there is no reason to expect differences between plants and animals. I'm pretty sure that most of you have some a priori, because the literature is full of a priori about this question. I have my a priori, and my a priori was wrong. My a priori, I was for the answer C, because for me, we had this kind of a universal threshold of divergence, mainly driven by recombination. If you're not able to recombine, well, you're not able to introgress. But we were wrong. Here, different hypotheses. I show you some a priori that you can read in the literature. So the a priori is that we can expect more gene flow in plants. Okay, you have a large body of literature on that with no data, etc. Just to say that, okay, maybe if I take this one, okay, the plants are plastic. If they are plastic, they can share everything. Or plants are immobile. They cannot escape. They have no choice to produce some hybrids, etc. So large body of literature about, okay, we expect more hybridization in plants. Still some articles explaining that maybe you can expect more introgression in plants, whatever. Here is the literature about, okay, we expect no differences. It's funny because some of the authors, like Mallet, who said we expect more hybrids in plants in 2005, 16 years after, actually, no, we don't expect the differences. And here is the literature about more gene flow in animals. I found nothing, actually. I found nothing. The idea is to, well, maybe we can just empirically test this hypothesis. So what we did is, well, in France, we do not have money, but we still have internet. So we can download some data from TBI. So this is what we have done. Here's a large phylogeny of plants with algae, bryophytes, dicotyledons, some monocotyledons here, some gymnosperm, etc. I took 488 pairs of population of species in 20, actually it's 21 genera. My criteria were very simple. We needed NGS dataset, whole genome sequencing, or RNA sequencing, or RADSEq, or exon capture. So we just needed some kind of large number of SNPs, actually. And I also needed at least 2 populations per species in order to have a kind of estimate of the level of heterozygosity. And we also need, well, at least 2 populations and 2 individuals. Okay? So by applying this criteria to NCBI, we found these 488 pairs of population species. Every time we downloaded the reads, we downloaded a reference from the 1KP project, the 1000 transcriptome project. We download the reads, we map the reads on the reference. So we apply the same SNP colors and the one used to call the SNPs for the animals, basically the same framework. And then we run the same model comparison, the same method. So it's quite comparable. The methods are the same between plants and animals. And this is what we found. It's the same result as previously. I just moved the relationship between divergence and because in plants we had too many points we start to visualize. But in green you have the relationship between divergence and the proportion of pairs supported by gene flow. So here basically all of pairs are supported by gene flow, and here we didn't find any pair supported by gene flow. In gray you can see the animals with this limit at 2%, and in plants quite the opposite of what of the a priori. So plant seems to stop gene flow more rapidly in plants. But actually, it's strong because it's a log scale. But here is actually we found an interruption of gene flow which is 3 times faster in plants than in animals. It's completely the opposite of what expected. Here we did model comparisons within models of gene flow. So here there are pairs of gene flow with a genomic homogeneity. So basically, this is a distribution of divergence for pairs of populations, distribution of divergence for pairs of semi-isolated species, and a distribution of divergence for pairs of species. Green plants, gray animals. So you can see here it's the intermediate stage in plants. This intermediate stage actually appears faster. You can detect more rapidly the effect of the first barrier in natural population. And it ends faster. For the same level of divergence, here you are still able to have gene flow in animals, but not in plants. It's quite funny. And after all, here we have a complete isolation for much lower level of divergence in plants than in animals. So here they are completely isolated, while in animals you can have populations or semi-isolated species. So one obvious question is, well, maybe it's a matter of geography. Maybe just because in your sample you take some very distant species, which is possible. So here I just plotted here the 953 sequence individuals. They're distributed in 21 genera, in Silini, Salix, PCA, Nepenthes, etc. So we have the GPS coordinate for each individual. It means that we can, here it's one zoom. For instance, here it's a zoom of, to show you what we did. It's in Helianthus here, the sunflower. Here is a zoom of the sunflower. The sunflower, in the Helianthus, we have 1, 2, 3, 4, 5, 6, 8 subspecies. And of course, we have multiple individuals. And for example, if you want to compute the geographical distance between, I don't know, Helianthus anus and Helianthus argopilus, well, we take the blue individuals, the one used for the demographic inferences, and we take the shortest geographic distance, okay, using the Haberstein method. And we did that for plants and for animals. Like that, we can get the distribution of geographic distances. In gray, the geographic distance in animals, here in plants, and here it's in kilometers. So they are not the same. They are significantly different, but not in the good direction. We have more isolation in plants, but plants, on average, they are much closer. So it's not an artifact of the geographic distance. We observe less migration in plants, even if plants are geographically closer than in animals. So here it's just a way to see, okay, the relationship between distance and gene flow. Well, basically, there is no effect, it's just noisy. So it's funny to see that there is no effect of the current geographic distance on introgression. But what is weird, it seems to have an effect of the ancestral geography. For pairs of plants and pairs of animals supported by ongoing migration, we computed the probabilities of models with ongoing migration since the time of split, and models with isolation in the past and ongoing migration. And basically, in animals, we find many cases of continuous migration. But in plants, it's the opposite. Apparently, it's more frequent to find in plants some period of past isolation. So maybe what it's not the effect of the current geography, but more the effects of past geography. During this period of isolation, you accumulate some species barrier, and during the secondary contact, you have a reduced migration. So I'm fully aware that what I'm showing to you is completely descriptive, actually. I have no answer why we observe an accelerated speciation in plants than in animals. Just a description of a pattern. And I'm pretty sure that when I will die, I will not have the answer because it will be complicated. But we have some ideas, of course. So the first one is, okay, what is the first difference between plants and animals? Well, the first one is that plants, they have a chloroplast. Not original, but they have chloroplast. And it's important because actually, the genetic basis of speciation are genetic incompatibilities. Basically, there are negative epistases between one locus of species A and another locus of species B. One specificity of mitochondria and chloroplast, there is a kind of, they're always involved in intragenomic conflict, which is the case of epistasy. So basically, plants, they are prone to have more intragenic conflicts because there are this conflict between the nucleus and the mitochondria, but they also have, in addition, this conflict between the nucleus and the chloroplast, which can bring new incompatibilities. Something to test. Maybe. Recently, in December, Sylvain Clément and Lucas Mariola, they published an article that maybe there is a contribution of selfing periods in plants. Paper, it's a theoretical paper. They show that if you increase the selfing rate, you also increase the probability to fix the barrier in your genome. And because plants tend to be more selfers than animals, it's very rare to find some selfer animal. So maybe this process of selfing, this period of increased selfing in plants, okay, it's also associated with an accelerated fixation of species barrier. Something to test. So this can be tested simply by doing the same work, doing the sigmoid between divergence and GFO, but between selfers and outcrossers. One project. Maybe also, for the same level of divergence in plants, we have more micro or macro chromosomal rearrangements. This rearrangement, actually, they decrease the probability to have introgression because they are barrier to recombination. So maybe this macro and macro chromosomal rearrangements, they are accumulated faster in plants and animals for the same level of divergence. It's something that we can only study recently because for that we need long-read sequencing, and long-read sequencing is not something that is so popular for the moment in natural population. So maybe we can see that in the future. It's funny because one argument of people saying that, well, we expect more gene flow in plants than in animals is because of the assortative mating. To say that in plants, there is no prezygotic barrier because they receive passively the pollen. Well, most of the time, the plants depend on the pollinator, and I don't know any animals that need the pollinator to produce the offspring. So maybe there's a contribution of the additional prezygotic barrier. I mean, here, the pollinator dependence. There is kind of specificity of the pollinator. Maybe it can increase the divergence. Here, maybe also in plants, we have quite different ecodemographic dynamics compared to animals. And it has been shown by using Fisher theory models, then this kind of cycle of adaptation and readaptation to different environments can increase the amount of barrier. So maybe in plants, because we have seen that in the past, they are more prone to be isolated in the past than animals. They are still they are continuously in contact. Stuff is maybe external fertilization in plants leads to stronger prezygotic selection, maybe because the ovule is targeted by different pollen from different species. So there is a kind of active process from the ovule to choose a good pollen. And maybe there is a kind of selection which is too strong, and a small level of divergence maybe can reduce the probability to being fertilized by a pollen which is not so distant, but too distant for the ovule. And something that we are tested in the lab is the haploid selection. Most of the barriers are recessive. It's something that we have deduced from the Alden rule. In the Alden rule, you know, it's when you have some hybrids which are XY or ZW. So in this case, they are with heterogametic sex, so that XY and ZW, they will have a reduced fitness compared to the homogametic sex. So basically, males in mammals have a lower fitness than females. What did we deduce from this observation? Is that, well, species barriers are recessive. Because if they are dominant, you do not expect any difference between males and females. So if species barriers are recessive, well, of course, we are diploid, but we are still able to produce some haploid. Spermatozoa, ovule, they are haploid. So maybe we have more genes expressed in pollen, which is more complex than spermatozoa, actually, than in spermatozoa. So maybe we have simply in plants the same number of barriers, but they are more expressed because we have more genes expressed in pollen. So it's something which is studied actually by Crystal Freys. She got an ERC from Europe to study a gradient of diploid phase in bryophytes. And the idea is easy. In bryophytes, you have a large diversity of the diploid phase. You have in the liverworts a very reduced diploid phase, but most of the time you're haploid. You have the hornworts, in which the diploid phase is longer than in liverworts, and mosses here with a bigger sporophyte than here. So the idea is to study this gradient of diploid phase. And for many pairs of Lagrange, many pairs of Mosses, and many pairs of angiosperm, which are the opposite, estimating the rate of introgression. If this process of haploid selection is strong enough, we are supposed to observe less gene flow in Mosses than in angiosperm. And if we have more genes expressed in pollen grain than in spermatozoa, it will mean that it can explain one contribution of this reduced migration in plants versus animals. So the idea is to do that along a gradient of this diploidy. There is a bug here, but we are supposed to see a picture. Just to do that by doing the same work as we did before. I mean, doing some genomic inferences about gene flow or isolation in different pairs of bryophytes, and to do that by a mathematical analysis. And it's funny because this part is already finished. Just doing some mathematical predictions about the rate of introgression as a function of the relative length of diploid and haploid, and it works well actually. If you have a longer haploid phase, you have a reduced gene flow. So why not? And okay, I think I'm over now. So I would like to thank all of the collaborators, of course, François Monet, my student, Zoé, Pascal, Émilie, Christelle, Yves, and Xavier. And it's over.
Do we have any questions for coming?
Thanks, really interesting. I was wondering if you looked at, or if you consider looking at this across different climatic zones, particularly tropical areas versus temperate and polar areas. And I'm particularly thinking that there's some quite old theories about temperate species having larger ranges, you know, Jansen's climate variability hypothesis and so on, which predicts faster speciation rates in the tropics versus the temperate areas, and, you know, kind of barriers to migration are more stronger in the tropical areas and things like that.
It would be very nice to do that. If I had the money, it would be a good idea. It would be to sample along a gradient, a latitudinal gradient. But what is already only available in the NTBI, there is a strong bias. We have mainly American pairs of species and European. So it's going to be hard actually with what is already available to test that. It requires a dedicated project. It would be funny actually to observe this reduction of migration as a function of the latitudinal gradients. It's cool.
Money always reduces, you know, our ability to investigate things, doesn't it? Money always reduces our, yeah. Any other questions?
Thanks a lot, Camille. That was great. Just this is a bit of an armchair kind of question, but if I get that correct, plants kind of evolve faster because they're split earlier, and it wouldn't be simply the trophic kind of relationship between plants and animals be something that explains it, because animals eat all the plants, most of them at least, and then so the plants have to evolve faster because otherwise they lose the kind of the game against the animals. Is that
what do you mean by
you can look at?
What do you mean by evolve? By the mutation rates?
No, I mean, you know, they seem to have, okay, if I get that correct, they split earlier, so you need less distance between the species or whatever populations. So that means in essence, they evolve faster. They have the ability to evolve faster. Is that correct?
Sure. To understand what do you mean by evolve faster? In what sense?
Yeah. Okay. I mean, if you have some property that allows you to differentiate earlier, faster, you are different, quicker, faster. That means your evolution rate should be also faster. And then in that sense, you outcompete your kind of predators, the animals, and therefore you increase. Has anyone looked at that?
Yeah, you mean that it's true that we here I only show the nuclear divergence, in order to see if there is a kind of molecular clock of speciation. But it's true that we do not have the dynamics of the threat. We do not have the rate of adaptation here. We do not have the rate of phenotypical variation. It's true that it's possible actually. Hard to have a straight answer. Possibly, one more hypothesis is that the plants have some traits evolving faster than animals. But I know nothing about that.
Oh yeah, really interesting talk, and I like that list of reasons why there might be a difference, and you've given a good example. Yeah, that list's really great. You've given a good example of testing this in plants, but it seems to me there's lots of opportunities to do that in animals. So, for example, the chromosomal rearrangements, like sort of reptiles, amphibians, and all the way back to Amphioxus, are really highly conserved in their chromosomes. But when you come to the mammals, it's all over the place. Dog's breakfast. So it'd be really good to see whether that's having an impact on the rate at which they can diverge, if that hypothesis is sensible. So we could test these in animals really easily. Quite a few of these actually. Some of them are internal fertilizers, some are external fertilizers. So really great list for fodder for people to test these ideas in animals. But I guess my question is, ploidy, does ploidy have any influence on this? I don't have no idea about the answer, but I tend to think that animals tend to have
it's true that
more diploid, and plants tend to have a lot of ploidy going on.
It's true that when I'm saying plants and animals, not fully honest, because it's a subsample of plants which are all of them diploid, and all of the animals are diploid here. And I'm only talking about a diploid organism. Yeah. So because it's complicated to make a comparison because most of the animals are diploid, the one I studied, and it will be complicated to compare gene flow between tetraploid plants versus gene flow between diploid animals. So here, focused on the diploid organisms. Yeah.
Okay.
But maybe, actually, it's one hypothesis that I didn't add to this list. It's not the current polyploidy level, but the variation, because in plants, it's very frequent to polyploidize and to re-diploidize. And maybe this dynamic, actually, because when you were polyploid and then you became diploid again, you have multiple copies, gene copies. And so you have an accelerated evolution because sometimes on some gene copies, you relax the selection on some genes. Maybe sometimes you have a sub-functionalization. So maybe this dynamics accelerates the rate of speciation, because for the same level of divergence, you have many more independent evolution of your paralogs compared to animals. So maybe this is one thing, not the current polyploid for me. It's more the current dynamic of polyploidization, rediploidization, subfunctionalization, neofunctionalization, etc. Maybe it can, for the same level of divergence, you expect to have more disturbed functional networks.
Yep. Thanks for that. On the ploidy question, do you have any plans to continue this sort of work looking at those higher polyploids? Because I mean, there are a lot of plants which have far higher ploidies than just diploid and tetraploid, in looking at up to like dodecaploids or things like that. So I guess I'm just interested to see whether this pattern sort of continues across far higher polyploids. So that's often something that's implicated in diversification rate increases.
I try to avoid thinking about polyploid because it's already complicated like that, and it will be nice, but because after that it will be okay, polyploid, but which levels of ploidy? Allopolyploid, autopolyploid. It's a mess, and then after that you have the exact polyploid with distant genes. But it should be actually funny to understand the rate of interruption of recombination within your polyploid. imagine you have sometimes you have because you can see polyploidization as a speciation event because you bring together different genomes into the same nucleus. Sometimes you can recombine, and recombination is migration actually. So recombining between your parental genomes is like having migration between two different species. So I think you can project the diploid models to polyploid in order to understand the interruption of recombination between subgenome A and subgenome B. But adding an additional layer of complexity, for me, it's complicated. But if you are motivated, you can work on that. Is
there any other questions in the audience? Otherwise, I'll check the chat and see if there's anything there. Look like there's any questions online. So if there's no other questions, please join me in thanking Camille for a really interesting talk.